share my slide. Can I do that?
Yeah. I have.
Share my screen
Yep.
here. Let's see, is that the right one? Yeah. So let me do this now. Okay, so what makes me crazy is that I always have this menu at the top here. But can you see my, is that a sort of the presenter view? Is that clear?
Yeah, it
Yeah.
looks
Yeah.
wonderful.
Great. Okay, so I'll just get started. So I titled the talk today "Cognition and Action in Parkinson's Disease" because that's really what we're interested in the lab, trying to understand the relationship between how the brain controls movement and how the brain controls higher cognitive processing, in particular decision making, and how that relates to movement control in regions of the brain, and how some of those processes go bad in certain diseases, particularly in Parkinson's disease, but also other movement disorders that you're probably familiar with, like dystonia and also Huntington's disease. So, by way of introduction, I like to use this slide, and it shows you really an example of the behavior that we measure in humans and also in animal subjects. And they're called saccadic eye movements, saccades, or rapid movements of the eyeball. And I can illustrate what these are using this work by Yarvis, who was a Russian psychologist back in the 60s. And if you aren't familiar with his work, I strongly encourage you to check out some of his work. He wrote a book a number of years ago, and it's just a really terrific, interesting read. He did a series of really cool experiments measuring eye movements in people and how they move their eyes while they look about visual scenes. And here's an example of something that Yarvis did. He presented them with paintings such as this, very complex visual images, and he monitored their eye movements while they looked about these visual scenes. And this is an eye position set of traces effectively collapsed over time, so that each line here indicates when the eye is moving very quickly in the orbit, changing position, changing the line of sight, what we call a saccadic eye movement. And then each point here is when the eye remains stationary in the orbit. Presumably, the participant is acquiring visual information, making decisions about where to look next, and so forth, right? So what you can see, I hope, is that in this pattern of eye movements here, this particular participant looked to these four locations here and up and down quite a bit. Can you see my cursor okay? Should I use the laser? I'm not sure if that's better. Is that better?
Yeah, that's perfect.
Although it doesn't move around as smoothly as the cursor does, so I'm wondering if I shouldn't use it. Yeah,
We
sorry.
can see the cursor as well.
I think I'm going to stick with the cursor if that's all right with you. If I can get out of this now. I don't know how to do that. Yeah, if that's all right with you, I'm going to stick with the cursor because it moves better. Can you see it? Yeah, okay, great. So what Yarvis did was as I was saying, they looked about these four positions here, up and down, and they correspond those positions to the positions of the faces of the people in this overlying image here. And the reason the pattern of eye movements looked like that was because Yarvis told his subjects, his participants, to report to him the ages of the people in the photograph. Had he given a different set of instructions, like estimate how long the visitor was away, he found an entirely different pattern of eye movements. So what this tells us is that we can use eye movements as kind of a window on internal processing or thought processes. And in this particular case, we see the influence of instruction, right? So there's something going on within the subject that we don't have access to other than through how they move their eyes around. So we study eye movements, we use eye movement measurements as a way to kind of make a behavioral assay of these internal processes. And as I said, in this case, you see the influence of instruction. Now, I can also illustrate using Yarvis some of the topics that we're interested in the lab. And they, over the years, have variously been called target selection, saccade, action or choice, and now decision making is the common terminology. And again, using Yarvis, you'll see that there are many visual objects in this scene. There's chairs, there are photographs, wallpapers, a window, different people, but eye movements can only be made one at a time. So there must be a process internal to the subject, internal to participants, that indicate which object out of complex scenes you want to focus on. You either want to tag that object for further visual information processing, or to identify it as a goal for a movement, or an eye movement, a leg movement, an arm movement, whatever kind of movement you'd like to make. The point is only one can be made at a time, so that internal process must happen where you select one target or choose one particular action or one particular saccade, or in other words, make a decision about what to do given this visual information. So these are the topics that we've been interested in. How does the brain do that? How does the brain identify one individual object of a myriad in the visual scene that we're looking at all the time? How does the brain make decisions about what to do, particularly when we have sensory information, and perhaps that sensory information is not always that clear. Sometimes it's a little ambiguous. How do we make decisions? What kind of information do we use in those circumstances? So in my laboratory, we really have a multi-method approach to try to solve some of these problems in neuroscience. And one of them, of course, is to use behavioral and computational methods working with humans, in particular humans with disease like Parkinson's disease, because we're interested in some of how these processes work in people with PD and how they might go bad or go awry in people with PD. And then to try to get a little bit more at the mechanism, how the brain does it, we engage in behavioral, electrophysiological, and computational methods in non-human animals, primarily in monkeys. Now, the beauty of studying monkeys is that they can engage in behaviors, cognitive behaviors, that in many cases are virtually identical to what we do in humans. So we can model the behavior in humans in monkeys very, very well. And so the brain mechanisms that give rise to some of these behaviors exist in monkeys and don't exist in lower animals like mice or rats, for example. I'm thinking of, in particular, the prefrontal cortex. There are regions of the prefrontal cortex that simply don't exist in species lower than primates. So to the extent that we're interested in understanding those higher cognitive processes and the computations that are being made in those cortical areas that are unique to primates, we need to study the monkey. And we do. We also, because the monkey is useful for many, many experimental approaches and questions, but not all of them, right? So the monkey is not always the perfect model, depending on the question that you're interested in. So if we want to drill a little bit deeper to get at mechanism, either pharmacological, biophysical, or synaptic circuitry, those kinds of questions we need to ask in different model systems. And for that, we use primarily mice, because we can capitalize on many of the novel techniques that are being used in neuroscience, primarily in mice, including optogenetics and chemogenetics. And we can talk more about those if you're not so familiar with those technologies. But they're not widely available for use in monkeys yet. But we, among other laboratories, are working hard to get some of those techniques working in monkeys so that we can do the same kinds of circuitry-based experiments using a much more complicated brain to understand a more complicated brain. So further by way of introduction, this is showing you a lateral schematic view of the Rhesus monkey brain, the monkey species that we study primarily in the lab. And what I'm highlighting here are regions of the cerebral cortex that are engaged in the generation of these saccadic eye movements, but also the visual stimuli that guide those saccadic eye movements. And so they're highlighted here. And the important point for this talk is that these cortical areas have direct projections down to a midbrain region called the superior colliculus. It's here on the roof of the midbrain, also called the tectum in lower species. You may have heard of that word before. That just means roof in Latin because it's the roof of the midbrain. The superior colliculus, in turn, has direct projections down to brainstem regions without which you'd be unable to move your eyes. So the superior colliculus is an extremely important structure because it's doing something really important, and we're trying to understand what, by taking cortical information en route to the brainstem for the generation of an action, in particular of eye movements. So we study the superior colliculus to try to understand what is the information processing that's going on in these pathways to generate, leading up to the generation of an action like an eye movement, an action choice. Now, we're also interested in the basal ganglia, which is a set of forebrain structures shown here schematically by this circle. It's obviously not anatomically correct, but it is part of the forebrain. And the important thing is that it gets direct projections from the same cortical areas, and then in turn has direct projections down to the superior colliculus. So we can study the basal ganglia, or nuclei within the basal ganglia, and compare the activity of those neurons in those nuclei with those recorded from the superior colliculus to try to understand what the processing is, the similar and different aspects of processing in these two pathways, in this direct cortico-collicular pathway and this indirect cortico-basal ganglia collicular pathway. So that's sort of the basic science reason that we're interested in these areas of the brain and that we focus on them. But there's also a clinical relevance to the kinds of questions that we're interested in. And as you probably know, the basal ganglia are involved in a myriad of diseases, both neurodegenerative, neurological diseases that involve motor control, like Parkinson's, Huntington's, dystonia, Tourette's syndrome, and a number of diseases, but also a number of neuropsychiatric diseases, including schizophrenia, drug addiction, likely also autism together with the cortex. So understanding what the basal ganglia do from a clinical perspective really can't be overstated, right? It has implications for so many disease processes. We really need to understand what this set of forebrain structures are doing. So that's the other reason why we're very much interested in basal ganglia, because we think our fundamental scientific insights and inquiries will have impact on the eventual understanding of how these brain regions go bad in diseases like Parkinson's. So please feel free to interrupt me at any time, by the way, if I say something that is not clear. As a colleague and dear friend once said, you know, I understand this stuff. The point is not to get me to understand it. The point is to get you to understand it. So feel free to ask me questions anytime to make sure you're tracking me. Okay. So a number of years ago now, there was a very famous discovery made by this Okihide Hikusaka and Bob Wirtz way back in the 80s. Many of you probably weren't born, not even twinkles in your papa's eyes, I imagine. But what they did was they trained monkeys to make very simple eye movements, what we call visually guided eye movements, such that the monkeys looked at a fixation point straight ahead, and then the target came on in the periphery, just a little adjacent, and the monkey was trained after a delay, that fixation point would be removed, and they'd have to look at where that spot was. All right, so really simple, what we call visually guided saccadic eye movement. And shown here are the eye movement traces now plotted against time, not collapsed, but plotted across time. So you can see the monkey's fixating, looking at the same position, and then there's a rapid change in position. By convention, leftward is downward. So he makes a rapid leftward saccadic eye movement and then fixates a little bit more. At the end of this trial, he gets a little sip of juice for reward, and this is what encourages the monkey to continue participating in this very simple task. Now, at the same time, Hikusaka and Wirtz, and we do this regularly. These are data that I actually collected, but they show the observation that Okihide and Bob made many years ago, is recording from individual neurons in one of the basal ganglia output nuclei called the substantia nigra pars reticulata. So there are two output nuclei of the basal ganglia. One is the globus pallidus internal division, and the other is the substantia nigra pars reticulata. This is that one, the substantia nigra. I'll probably call it the nigra for short. And each tick here indicates an individual action potential recorded from a neuron in the nigra. And each row of ticks indicates a trial in which the monkey made one of those eye movements. neurons, right? So there are a bunch of trials here, and there are actually a bunch of saccades here, but they're all superimposed because that behavior is very machine-like in the monkeys, in people too. And then this activity right here is showing you essentially the average activity over all these trials. So what I hope you can see is that these neurons have a very high discharge rate. They spike discharge action potentials at very high frequencies, upwards of 100 action potentials per second, sometimes even higher. And then around the time of the eye movement, they show this very little itsy bitsy pause, not a whole lot to write home about, but a slight decrease in activity. But interestingly, if you have the monkey make exactly the same eye movements now, but to a stimulus that's no longer present. So in other words, the monkey has to remember where that spot was, right? So now the eye movement is guided by memory as opposed to vision, right? You see a profound modulation in the neuronal activity, right? It pauses, it decreases its activity a lot more. So this was sort of the first evidence that basal ganglia is not just involved in any kind of movement, but specifically with movements that were what we called internally generated, in this case, generated by memory rather than by an external stimulus that drove the movement. And this is consistent at the time with what people understood to be part of the Parkinsonian syndrome, and that is they had trouble generating internal movements, whatever internal movements meant. We really didn't understand what that was, right? But memory is an example of an internally generated movement. Now, the other thing I should point out is that these neurons are thought to contain GABA, the inhibitory neurotransmitter. So when they're active, they're providing a tonic level of inhibition onto the superior colliculus through their direct projections. So that when they decrease their activity, they remove that inhibition, and the superior colliculus can then discharge action potentials to drive an eye movement, okay? So that's how this circuit was understood. But it wasn't, again, just any old eye movement. It was specifically this eye movement guided by memory. So we followed up on these experiments a number of years ago now, and a number of years ago since that initial observation, by doing a very simple experiment where we thought we would manipulate the substantia nigra neurons, their activity, and see whether we could preferentially affect movements that were guided by memory rather than movements that were guided by vision, so these internally generated movements. So we had monkeys make saccadic eye movements, 2 targets located all around the screen, so up and down, left and right, and then the 4 oblique positions here. And we had the monkeys do that in a visually guided condition, so the stimuli were present that they had to look at, or the memory guided condition where the stimuli were flashed and removed and they had to look where it was, okay? And the blue is showing you the traces when we introduced electrical stimulation, and the black shows you traces when we did not, when there was no electrical stimulation. And we randomly interleaved all these trial types, so the monkey couldn't really predict what was going to happen next. Okay? And I hope you can see here that for the visually guided eye movements, the monkey was virtually unimpaired. He could still make the eye movements, whether or not the electrical stimulation was manipulating the activity of those neurons. Whereas in the memory-guided eye movements, the monkey had profound impairments. He was effectively unable to make eye movements into the contralateral or opposite hemifield. So altering the basal ganglia output altered the memory-guided eye movements, leaving the visually guided eye movements relatively unaffected. Now, the other thing that was really important about this experiment, and that was followed up in the second experiment down here, was that with electrical stimulation, we could activate the neurons at different times during the task. So we could stimulate when the visual stimulus appeared, presumably when that spatial evidence was coming into being encoded in memory. We could stimulate during the delay period while the monkey was remembering the location before he actually made the eye movement, or we can stimulate during the movement itself, right? And see what's the contribution, what time period is the best contribution of this change in activity. And what we found, in fact, was the effect happened when we stimulated during the movement, but as this example shows, it also occurred when we stimulated during the delay period. So even well before the monkey made the eye movement, changing the activity of the basal ganglia changed the monkey's ability to make a subsequent movement when it was guided by memory, not when it was guided by vision. So far, so good? Yeah, great. So this led us to a hypothesis that the availability of this visual information really changes the contribution of the basal ganglia to movement, and more importantly, to the events that precede movement, like spatial memory or specifying an eye movement, preparing an action, or making a decision about whether or not to look at something. And this also reminded me, because I had a long-standing interest in clinical symptoms of Parkinson's disease beginning as a graduate student, in this phenomenon called paradoxical movement. Now, many of you may be familiar with this. It is a phenomenon that's seen in Parkinson's disease, wherein people with PD, and if we were together, I would illustrate this for you, so it might be a little hard on video, but you know, PD is very characteristic, right? That people have a stooped posture and a kind of a shuffling pattern to their gait, right? That shuffling pattern is called festination. Now, if you draw high contrast horizontal lines on the floor, and you ask a person with Parkinson's disease to step over those lines, you can increase their stride length, right? So you can make their gait almost not perfectly normal, but normalize it, right? So what this tells us is that the locomotor machinery in the spinal cord that drives the gait, the walking pattern, is intact, right? Because it can be initiated when there's visual information, right? But in the absence of visual information, they have trouble doing it. They can't get the gait pattern right. Now, it turns out that the same phenomenon is seen in eye movements. So if you ask a person with Parkinson's, at least in the early stages, to make a saccadic eye movement to a visual target that's present, that saccadic eye movement looks like a healthy controlled saccadic eye movement. The velocity is relatively the same. The amplitude is relatively the same. But now if you have them make a memory-guided eye movement, you see sometimes this festination. They make small eye movements out to the end, or they just come up short. They're what we call hypometric. And they're also slower in their velocity. So they have the symptom of what we call bradykinesia, right? A slowness of movement. So you see all of that, all of their segmental problems are recapitulated in the oculomotor system. So this phenomenon that we saw with the stimulation reminded me very much of that Parkinsonian symptom. And I asked a very simple question, or I should say first, you might imagine how alterations in the discharge of neurons that look like this could give rise to a symptom like that, right? You change this activity pattern and you might impair the ability to make a memory-guided saccade, right? But it required me, or it made me think of, well, I wonder if this phenomenon extends to cognition, you know, to a decision-making problem, or if it's really just a motor problem, right? A paradoxical movement, or is there such a thing as paradoxical decision-making, perhaps? So could we look at participants and ask them to make decisions with clear sensory information and with ambiguous sensory information where they had to rely on their memory information? And do we see preferential problems in people with PD if they need their memory to make their decisions, right? Pretty straightforward, right? So here's the task. Now, I don't know that this is going to work because the internet and Zoom, and so it's stopping. In the experiment, it doesn't actually stop like that. So that's an internet or a Zoom problem. But I hope you get the sense that these patterns are dynamic, right? They're flickering. And what I hope you can also get the sense of is that they're made up of a bunch of dots, and the dots have some coherence to them. So in this particular case, the dots are sort of paired and they're kind of tilted to the right. Right? Is it your left? No, you're saying it is right, right? Yeah. Okay, good. Yeah. Whereas this one, they're not really well paired, and you don't get a clear sense of orientation, right? So if that's not true, I recommend consulting with a ophthalmologist and possibly a neuro-ophthalmologist, because this is something one should see, right? There's an orientation signal here, okay? So what we ask is for the participant to tell us, do you see it tilted to the right or do you see it tilted to the left? And we vary the difficulty, right? So here is a relatively easy decision. Yeah, it's tilted to the right. Here's a really hard one because there's really no orientation information. And we can do all of the coherences in between to get what we call is a psychometric function shown here. And this is where we plot just the proportion of positive or say rightward choices as a function of the orientation strength, the signal or the coherence. And 0% coherence was that one that I showed you on the left, where there's absolutely no orientation information. So what the subject does is they choose left and right 50% of the time. They're just guessing, right? Which is what they should do because there's no information. Okay? When it's very strong rightward information, they choose rightward all the time. When it's very weak rightward information, they choose rightward almost never, right? So the decision, the choice of the orientation is really determined by the strength of the sensory evidence. Right? When the sensory evidence is weak, people randomly guess. Right? Okay. So now a very talented postdoc in the lab, Alessandra Perugini, she's now at the University of Bristol. She did a really interesting experiment with people with Parkinson's disease. She presented them these glass patterns, that's what these patterns are called, but she presented them with different colors. So in one case, some of them were red and some of them were green. And she varied the coherence, just like I showed you a moment ago. But the trick she did was for some of the glass patterns, say the red ones, she varied the probability that the left orientation would occur more often. She didn't tell the subjects this, right? They had to learn these statistics implicitly. She randomly interleaved all these trial types. So what she had was on a bunch of trials, say for the red glass pattern, 80% of them would occur left, 20% would occur right. And then for the green one, 50% would occur left and 50% would occur right. So the idea is by varying that probability information in this sort of feature-specific, color-specific way, she could ask whether people used memory information, that statistical probability, when faced with sensory uncertainty. So if the sensory information was ambiguous, if the coherences were not very clear, would they fall back on those statistics that they had learned, right, that they were learning? And sure enough, healthy people do this. So what you can see is what I showed you before, the gray. This is with the 50-50 probability. Here is now, the black is showing you when she varied the positive orientation, it occurred more often than the negative. Let's say positive is right, negative is left. And what you see is even in the 0% coherence condition, when there's no orientation information, they started to choose positive more often. So they were using that memory information. Likewise, if she introduced what we call a prior, a statistical prior, in the other direction, participants chose the other way more often, even though there was no sensory information in this display. And you can see they did that primarily for the sensory conditions that were difficult, whereas for the easy ones, they didn't really do anything because they had the sensory information, right? So she now had a task where she could manipulate memory information in a decision task to see whether people with Parkinson's were impaired at this. And sure enough, it turns out that people with PD are impaired at this. So here's an example now from 12 patients, same task. And what you can see is these curves are all overlapping here. So the participants simply did not use that prior probability information to guide their decision making. They just couldn't do it. Whereas age and sex matched healthy participants had no problem showing decision biases in this condition of statistical probability changes or changes in priors. Right? So that was really interesting. So we thought, okay, you know, maybe this paradoxical movement does extend to decision making, that there is this common phenomenon of when sensory information is absent and patients have to rely on other information like memory, they show impairments. Well, one possibility is that it could be because they were on their medications. So we know that dopamine plays a role in some of these cognitive processes, also reinforcement learning. So it could be that just because they were on their meds, they were showing these problems. Well, Alessandra went and took 15 more patients, different patients, and had them on and off their medications, and she saw the same problem. So based on this, we concluded that the impaired memory-based decision-making was independent of the dopaminergic tone. Whether they had their medicine on or off, they still showed the same problem. So probably not something related to reinforcement learning, right? And in fact, she compared the effects of dopamine, their medications, on their movement and on their cognition, which I just showed you. And she found that the dopamine did impact their movement. So when they were on their meds, they actually scored better on their motor assessment, the UPDRS. So we could say that the dopamine was working, right? You might say, well, of course you got no difference. Maybe they've lost so much dopamine, you give them their dopamine medication, it doesn't work because there aren't any cells left. Well, it is working because it improves their motor scores. It just didn't improve their decision-making performance. And she just correlated their ability to use the prior information with their motor score, showing better movement here, and there's just no correlation. So this was sort of dissociable. The dopamine affected their movements, made their movement better, but did not make their ability to use the memory information to guide their decisions. And another thing she did, which was really interesting, was she looked at another patient type with basal ganglia disease, but didn't involve degeneration. It's called dystonia. It's a focal dystonia. You may know it as some people have writer's cramp, or they have spasmodic torticollis, which is a problem in the neck. It's a co-contraction of agonist and antagonist muscles, leading to sort of deformations in posture. So she looked at people with these focal dystonias that involve the basal ganglia, but don't involve degeneration, and don't, like Parkinson's, and don't involve dopamine. So these people are unresponsive to dopamine treatments. And sure enough, she found the same impairment in this basal ganglia disease population. So even people with focal dopamine unresponsive dystonia are impaired at using memory information to guide their decisions. And again, it didn't correlate with their motor score. So it wasn't like they were worse if their motor symptoms were more prominent. They seemed to be dissociable. So she also did some other experiments to rule out that it was a learning problem. She simply told people what the priors were, and they still showed impairments. So we're pretty confident that this phenomenon of paradoxical movement actually extends to decision-making, that it's a kind of paradoxical decision-making. And as a result, we think that some of the circuits that underlie this cognitive impairment might actually be at one of the core things that involves or produces the motor symptoms in people with Parkinson's, that these things might actually be related. So one of the things we wanted to get at, is somebody paying attention to my time? Because I realize I'm not.
You're good on time, Dr. Basso.
Okay, so just give me a heads up when I need to wind down, because I could go on for days. So, okay, so then what we wanted to do was try to understand the mechanism of this process a little bit more. And so what we did was we teamed up with a colleague at UC Davis, who's an expert in computational models and decision-making models in particular. And we applied a pretty popular decision-making model. We fitted this model to our data to try to understand what is the mechanism that underlies the problems that we see in people with Parkinson's. And so this model is super simple. It's called the drift diffusion model. It's an example of a broader class of models called sequential sampling. And all it means is that you have noisy sensory evidence that occurs on sample time points. So there's momentary evidence that has to be accumulated over a period of time. And that when that net evidence, which is essentially a difference in evidence for one choice or the other, when that reaches a threshold, you make that choice. So for example, we have this noisy sensory orientation evidence that starts midway between these two boundaries, this being a right choice, this being a left choice, starts midway, and it starts creeping up toward one of the bounds. When the bound is reached and crossed, you make a decision and say, it was rightward, okay? So very simple model. We have some understanding in the brain of what brain areas are involved in this, but not all. It's still an active area of research. And the terminology is, this accumulation of evidence is called the drift rate. And it's really determined by the strength of the sensory evidence. So if you have really strong sensory evidence, the drift rate is going to be fast, and it's going to reach the bound more often than it goes to the opposite bound, making a mistake, right? So you're more often to be correct, okay? If the sensory evidence is weak, this drift rate will be slower, and you'll sometimes just by chance go to the wrong decision and make a mistake, right? So reaction times are longer, the speed with which you make a decision is longer, and also the probability of error is increased with less sensory information. Makes sense, right? Very, very intuitive. Okay? Well, in this model framework, prior information can affect decisions in two possible ways. One way is by changing what we call the starting point. So where you start to accumulate evidence actually gets closer to one of the bounds. So the amount of evidence that you have to accumulate is actually reduced, right? Because you have this more frequent orientation information coming in, so you don't have to acquire as much sensory information to make that choice. Okay, so that's one way. The other way is to make that drift rate go faster, right? It's essentially increasing your perceptual sense of orientation. You think there's more orientation than there actually is, right? So that's the second way. So we use this model to fit the data, we fitted the model to the data, I should say. And what we found, interestingly, was that people with PD were actually able to adjust this drift rate offset very well. So they could tell you, well, they couldn't tell you verbally, but the brain could tell us by these model parameters that something was different about these stimuli, right? Because they changed the drift rate offset here. So this is just plotting the drift rate offset for the first half of trials and the second half of trials. And you can see, because we assumed that it took time for them to learn about the prior. And you can see that they adjusted their drift rate for the unequal prior condition. But for the equal prior, there was really not much change. But what they failed to do, or where they were impaired, is in this starting point. So the adjustment of the starting point, interestingly, they adjusted it kind of in the opposite direction. So they went closer to the other bound, which would cause them to make more mistakes, essentially, right? And when it was there, it got kind of stuck there. They just couldn't change it. They couldn't flip it over the other way, right? Because the way to have done this would have been to flip this over to this side. So you bring your start point closer to the more frequent choice, and you bring your drift rate offset to be faster, so you make more frequent choices more often, right? Does that make sense? Sort of? Yeah, great. So people with PD were impaired at this. And they showed this impairment at the starting point, and they were able to adjust the drift rate offset, but they probably adjusted it too much, actually. But that's another story we can talk about later. So this led us to the conclusion that people with basal ganglia disorders could really learn the priors, but they couldn't actually express this bias in their decision making because they had this impaired ability to adjust the decision thresholds or the amount of evidence that they needed to accumulate for a decision, okay? And this suggested that the inhibitory output, that basal ganglia, perhaps superior colliculus pathway, might be involved in regulating decision thresholds by altering this starting point of evidence accumulation, altering how much evidence needs to be accumulated for a decision. So we then next wanted to know, well, where and how are decision thresholds determined? We really don't know. There's not a lot of study on this at all. And so we engaged in one of the first experiments to try to get at how are decision thresholds created in the brain? How does the brain know when to make a decision or what decision to make? So we reasoned that our superior colliculus, that midbrain structure that we like so much, because it receives input from the basal ganglia, also receives input from the cortex. It's a nice station to get all of this information and integrate all of this information. It's involved in eye movements, producing action choices. We know it to be involved in attention. You shift your attention when you shift your eye movements. So possibly this pathway, and the superior colliculus in particular, might be involved in adjusting these thresholds. So another very talented former postdoc in a lab, Dr. Trinity Crapes, did a really nice experiment where he tested this hypothesis, now using monkeys. And so he trained monkeys to make a simple decision task, what we call a yes-no decision task. He had monkeys fixate on a spot in the middle of the screen that they were looking at, and then two possible targets appeared, a green one and a red one. And then after a period of time, one of these glass patterns appeared. And in some cases, it had orientation information, pretty strong orientation information. In other cases, it didn't, or it had weak orientation. And the monkey had to respond. If it saw orientation, look to the green spot. Say, yes, I see orientation. If it doesn't see orientation, report no and look at the red spot, okay? And so he also randomly interleaved these two positions, the red and green. So the report was really a yes or no report. It wasn't just a left or right report, right? That's important because in the superior colliculus, there's a very strong map of spatial locations. And one could say that, well, if you see activity there, it might just be because he's more likely to look left. It has nothing to do with the decision, but it has to do with sort of a motor bias, right? So that's why he flipped the targets to rule that out, okay? So then what he did was he presented a balanced block of trials, which we call 50% of the trials had no coherence. So the monkey's answers were always no here. And then the other 50% were yes trials, in that they had some amount of coherence. This very easy, this rather difficult, but still the answer is yes, okay? And then what he, excuse me, what he did was he primed the monkeys. He gave them these prior probabilities. And in one case, he said, okay, yes is occurring on 85% of the trials, and no is going to occur on 15% of the trials. We call that the liberal priming block, right? Because he gets the monkey in the mode of saying yes more often. Or on other days, he put the monkeys on what we call a conservative priming block. He did just the opposite. So the probability of saying no is 85%, and the probability of saying yes is only 15%. So now the monkey's in a groove of saying no, he's more conservative, okay? And then what he does is the balance block again, okay? And the same 50-50, and asks what happens to the monkey's behavior? Does he shift his choices based on those prior probabilities, just like we saw in healthy people? And what does the brain do? Does the brain reflect those changes in decisions that the monkey makes in this task, okay? So now I'm going to give you a little introduction to signal detection theory. How many of you are familiar with signal detection theory? Maybe? None? No? Okay, so good. I'll give you a quick overview of this. Whoops, let me back that up. So imagine there are two distributions. This is a simple task where we ask the monkey to say yes or no. So you can imagine that in the brain, there's a representation of the orientation signal somewhere. And there's a distribution of that sense of orientation. So some neurons are firing a lot, some are firing a little, but across a population, there's some representation of that orientation information. And there's other parts of the brain, essentially, that that representation doesn't exist, right? So you can imagine two distributions, one in which there's a representation of, yes, I see orientation, and there's another that says, no, I don't see orientation, okay? And in signal detection theory, this is used a number of years ago to for sound localization. Do you detect a sound or do you not detect a sound, right? And it's traditional psychophysics. And what you can imagine is if there was a choice or a stimulus drawn from this distribution, and your decision criterion was right in the middle here, and you were going to say yes to everything that came to the right of this criterion. So if something was drawn from this distribution here, you would say yes, you saw orientation, right? And if that were the case, came from this distribution, it's to the right of this criterion, that would be called a hit, because the stimulus actually appeared, and you said yes, it appeared. Now, what you'll see is that this noise distribution, where there's no orientation signal, kind of overlaps here a little bit, right? Because there's noise, right? It's not a delta function, but it's a Gaussian. So some of this distribution actually falls to the right of this curve, right of this set of distributions. So if the signal, or if the stimulus now was drawn from this distribution, the noise distribution, so there was no orientation signal, but you still said yes, because you perceived it as to the right of your criterion, we call that a false alarm, okay? Makes sense. We said it was there, but it wasn't. It's false alarm. And the same thing happens now for your no decisions to the left of the criterion. So if I draw a stimulus from the noise distribution and you say, nope, no orientation, that's a correct rejection. However, if I draw from the orientation distribution and you say no, because it's to the left, your interpretation is to the left, that's a miss, because it was there, but you said no. Okay, so those are 4 different trial types that we can get in this type of task, either a hit or a miss or a false alarm or a correct rejection, right? And signal detection theory gives us the math to tell us how decisions are changing. So for example, if your criterion now gets more conservative, right? You move your criterion to the right. Now, when I draw from this distribution, right, it's going to be really high levels of signal that give you a hit, right? And even levels here, you'll say no for, right? So when you get more conservative, what happens, is that your hits will go down, right? Because there are many fewer hits here. You're going to be saying no a lot more. And your false alarms will go down, right? Because now none of this noise distribution falls to the right of your criterion, right? So your hits and false alarms are going to go down. Now, if your criterion becomes more liberal, right, and you shift to the left, you're saying less more often, now what happens? Both your hits and false alarms are going to go up, right? Because there's more of both of these distributions to the right of your criterion where you're going to say yes. So far, so good?
Hey, Dr. Basso, we're getting closer to 6, so not sure how much is left, but we might need to have room for questions at the end.
Yep, I'll wrap it up. Yeah, I'll wrap it up. So the final answer is, I can end it right here. Using this signal detection theory method, we found by recording from superior colliculus neurons that the neurons in the superior colliculus actually reflected these changes in the decision criterion and told us where the monkey placed his decision criterion. So we think that the things that we're seeing in people with Parkinson's disease, the cognitive impairment related to decision making, is actually mediated via this substantia nigra pars reticulata superior colliculus pathway that's reading out decision criteria, what you decide, what sensory evidence you need to say, yes, I want to choose that was right, or yes, I want to choose that was left. Okay, so I'll leave it at that. I'll summarize that way. We think it's this low-level brainstem area, really, that's involved in this higher cognitive process and might actually underlie some of the cognitive impairments that we see in Parkinson's disease. Okay, how about that? I'll just wrap that up.
Awesome. Thank you so much, Dr. Basso.
I hope that made sense.
No, great.
Can I stop sharing? Yeah, there we go. Great. All right.
So I know we're at 6, but if anyone has any questions for Dr. Basso, I think we can stay an extra maybe 5 to 10 minutes.
Yeah.
We have one question set up already, so unless anybody has a great question, we can start off with that. Okay. Yeah, and also if you have questions, just put them down in the chat and we can read them.
Yeah, yes, let me see, yeah. So perfectly crystal clear, no questions, right?
So our first question is from Sasha, and it's a fun question. And so it's, if you weren't a neuroscience and behavioral researcher, where do you think you would be, or what would you be doing?
I don't know, on Wall Street making money. I don't know. Yeah, I wanted to go to medical school, to be honest, when I first started, but I very quickly got bitten by the research bug. I took a class in what at the time was called psychobiology. Today we call it behavioral neuroscience or systems neuroscience. And I just thought it was the coolest thing since sliced bread. And the thought that, I could be in the lab discovering things that in a moment I would know something that no one else in the world would know at that moment was just so thrilling to me. The thought that I could do that on a regular basis, was just so thrilling to me, particularly about the brain, the most complicated organ in the universe, really. Um, it was just too exciting. The thought of, being a physician was more like, okay, I'm probably going to do the same thing every day. I'm going to see a lot of the same things. Yeah, there'll probably be the occasional, exciting case here or there, but pretty much research is different every day, right? And that really thrilled me. So I don't know what I would do, to be honest. I can't imagine that. I suppose I should take some time to do that, but I would like to make more money, though. Academics is not for people who want to make a lot of money, that's for sure.
I love that. I also wish I could make more money, but being kind of hurts that. So,
Right.
does anyone have any questions before we, any questions for Dr. Basso?
Well, feel free to shoot me an email if you have any questions. Anytime. I'm happy to help or answer anything I can. Mm-hmm. Even if, if it's advice about grad school or med school or whatever, I'm happy to give a hand if I can.
Yeah, we can definitely send out the email.
Yeah, no problem. No problem.
Awesome. Thank you so much, Dr. Basso. If everyone give a virtual round of
Thank you.
that was just wow.
Thanks. So another time I'd like to come back and hear about what all of you are doing.
Yeah, we'd love to keep in touch with you. And thank you, RT, again for inviting a wonderful guest speaker today.
Thank you, everybody.
Thank you, Dr. Basso.
Thank you.
Bye-bye.
All right, everyone else, have a great rest of the quarter, and we'll see you later. I shall end the recording.